Hi Megan,
Hello,
how are you doing?
I'm doing great. Happy to be in California.
Happy to have you here. It's exciting. So you're Megan. You're an evolutionary biologist, and you're currently assistant professor at the University of Toronto. You're interested in defining the principles that shape species geographic distribution, and more specifically in how ecology and evolution interact to define the species distribution. So you've worked in the eco-evo field for about, 10 years now since your
Yeah, I think
since
so.
the beginning of your PhD in 2012. So what drove you into this field at the very beginning?
Yeah, so I initially thought maybe I would go into like biomedical sciences. I wasn't really sure when I started my bachelor's degree. And in the intro bio series during my undergrad at UC Santa Cruz, I just had the most fantastic professor who just loved plants. And this is Jen Yost, who's now at Cal Poly, and her enthusiasm for teaching the plant half of the plant and animals course just like totally caught me. And I asked her about volunteering in her lab. Turns out she was just a PhD student at the time. But I'd say it was really her enthusiasm and then the mentorship of other professors during my undergrad that really got me started on studying plant ecology and evolution.
So it's interesting because it's funny because I don't think I've heard that a lot from eco-evo people, where most of it was based on their will to do something about climate change, and yours is really focused on the science and being excited by the field, and people taught you about the science. That's pretty cool.
I love the problem solving, or like the puzzling, trying to explain why something is the way it is. And it's great if we can take that and apply it to climate change. But my initial interest was just sort of, why are plants the way they are?
And so I found it funny that during your PhD you went to study a plant that I didn't know about before reading reading your thesis. How do you say that title? Which is Clarkia pulchella.
Yeah,
right?
Clarkia pulchella.
So is that a plant species from California? Is it a native plant from California, or where?
So there are many species of Clarkia in the genus Clarkia in California. But this particular Clarkia only occurs in sort of the interior Pacific Northwest. So I did my PhD in Vancouver, and I wanted to find a study system that was an annual plant species so that I could grow it quickly and that I could drive to in a reasonable amount of time from Vancouver. And so I looked at lists and pictures, and Clarkia pulchella ended up being the one I picked. So I went out.
What was the main driver for picking it? So you say,
I mean, it was really a
plants.
practical search. Like, I wanted something where it was an annual, it didn't have super strong seed dormancy, so that I wouldn't have to like deal with that when trying to do experiments with it. Something I could hand pollinate easily to generate family structure and make seeds for experiments. And then something with a geographic range that was limited enough that I could travel all across the range within a single field season. And so through a process of elimination, it ended up being Clarkia pulchella, and it's ended up being a very good study system. It's a lot of fun.
So that's interesting, and I think it brings us to what are the kind of tools that you've been using, because it seems that you've chosen the system to be able to grow it in the lab and do things yourself on it, as well as going on the field and doing sampling on the field. So what kind of tools have you been using in this system?
Yeah, so in this system, we've used a bunch of approaches. So they're like most of the West Coast, the Pacific Northwest has great herbaria and great plant collection resources. And so we've worked with measuring phenotypes of specimens in the herbarium, and then examining the relationship of those phenotypes to climate across the range. We've done field transplant experiments. We've manipulated the pollination environment in the field, and we've done some...
Sorry, how do you do that?
By putting little tents over patches of the plants to keep pollinators off of them. Yeah, to see sort of this plant is mixed mating, so it can outcross or it can self-pollinate. We wanted to see if how well it could self-pollinate varied between populations in the range.
Okay, interesting.
Yeah.
And so you've, after that you've moved, no, you did a postdoc before that. Did you stick to your model system of your PhD, or did you change the model, like going to Arabidopsis or another plant model system?
Yeah, so I switched during my postdoc. It was a little sad to leave the Clarkia behind, but it was...
Are you still working on it a little bit?
I've come back to it, yeah, so it's not left behind forever. But I was really excited. So Clarkia, the Clarkia pulchella system was one I developed by myself and worked in mostly by myself during my PhD. And so then I was really excited for my postdoc to join a big team of people all working on the same system, which is probably another system that maybe you haven't heard of,
That's very
which is Streptanthus. And so Streptanthus is a genus of plants mostly native to California, and they occupy a variety of habitat types across California, including huge elevational gradients. And so some of my work has been on like life history evolution in that genus, and so that's what I switched to in my postdoc.
Okay. And so now that you're in Toronto, what did you? So you said you switched back to your former model system. Are you still using both of them? What kind of new, what's the new field that you're trying to develop with your lab right now?
Yeah, I mean, I think that's a really good question and and sort of an open question for my lab. So I'm carrying forward work in both Clarkia and Streptanthus, but doing field work on the West Coast is not going to be logistically the easiest thing for graduate students and undergrads in my lab. So this summer, I'm hoping to do a lot of exploring in the area around Toronto and also find some local systems to start working in. So there will be some new systems in my lab, I hope, in the next couple years.
Okay. And what kind of tools have eventually been developed in the last 10 years that allow you to now, basically, are you now able to say this population will be able to spread out or will shrink in geographic space in the next 10, 20 years? And what kind of tools have allowed you to develop this kind of understanding?
So I think the question of what limits ranges and whether ranges will expand is still an open one. Despite like a lot of good work on that question, I don't think we have like a good understanding, and certainly not a good like predictive ability on that front. But I think we're slowly accumulating a large amount of work done in natural systems, moving plants around, seeing how they perform, moving them beyond their range edges and seeing if they survive, and getting a better understanding of the phenotypes and genotypes that'll be appropriate in new areas beyond the current range of species. And so I think we're on our way, but I think there are questions that just take like a lot of hard, tedious work, ideally field work, and it'll be exciting to see how new approaches, genomic approaches, sort of inform our understanding of what limits ranges. But I think there's still a long way to go.
Okay, interesting. Do you feel there has been a discovery in the last 2 years that has been really groundbreaking in this matter, in the interaction between, for example, genomics and the geographic area? Or...
Yeah, that's a good question. I think, so maybe it's not a groundbreaking discovery, but I think something that my group's been working on also, in addition to sort of what limits ranges, is what exactly are populations adapted to within their range? And this is an important question because if a range is limited because populations haven't adapted to the area beyond it, knowing what they are currently adapted to in their local range can inform our understanding of what might happen outside the range.
Is there a specific parameter that's like the limiting factor for the range? Can we see it like that, or is it probably too simplistic?
I wish it was that easy, and it should be, right? But it's not. But some of the work we've done, so we've been working on like synthesizing data from all of these experiments people have done, moving plants around within their range. And one of the findings that is maybe not surprising, but that is strong in general across studies, is that temperature is a huge driver of local adaptation. Okay. So, when a population does well in its home site and does poorly somewhere else, a huge amount of that performance variation can be attributed to temperature.
Like the average temperature over the year, or the maximum, the minimal, or the variability of it? It's
hard to tell because all of those are so correlated, but even just using average temperatures during experiments and comparing them to what have populations experienced historically, we can explain a huge amount of variation in performance among populations. So it's not shocking that temperature is important, but it's nice to see that coming out as a generalized result.
Yes.
And so then I think the next big open question is, okay, that explains about half of the performance variation we see.
It's huge.
What's making up that other half? And I think that's a interesting open question. We tried looking at precipitation, couldn't pull out a signal. Tried looking at biotic interactions, we couldn't pull out a signal. Those might be power limited, but it'll be interesting in the coming years to figure out exactly what's making up that other half of local adaptation.
Could it be the sunlight limitation?
Yeah, I mean, it could be something like that. Like they're either at large scales day length, at smaller scales sort of microhabitat characteristics. And the thing about temperature is temperature data is available at such a massive spatial scale. It's easy to work with. And nothing else is like that.
It's fascinating to see that temperature is explaining 50% of the range distribution of species. That's quite important, and like giving the main change in climate that we're gonna see is an average temperature increase overall.
Yeah.
So it's kind of easy to feel, at least from a very naive perspective, how these studies can be implemented to predict future landscapes.
Yeah.
Is that where you think the field will be going in the next 10, 20 years, or is that what the kind of impacts it will have?
I'm always a bit of a prediction skeptic, but I do think that we can safely say that the changes in temperature that are already happening and that are coming are going to have massive effects on fitness and performance in natural populations. They'll probably have indirect effects on interactions between species, and that sort of maintaining an equilibrium with biodiversity is probably going to mean large range shifts or movement of lineages within the range, and sort of large reorganizations of biodiversity. So I do think that there are big implications for sort of how nature is going to respond in coming years.
Okay, that's really fascinating. I have so many other questions, but I think we'll have to end here. Thank you very much, Megan.
Thank you so much.